know we are still getting settled down. So I'll be talking about flow cytometry today and in the context of HLA typing, HLA analysis, and primarily what we're discussing is the HLA antibody assays. I bring you warm regards from the hospital where I work, which is in the heart of Delhi on Pusa Road. And of course, this is my lovely team back home, which helps me to collate the data and share stuff with people. And this is what I have done. I have outlined my talk, giving you a few cases to start with. We'll talk about DSA, and I think probably since a lot of it has already been discussed, I will not really delve into details of that. We'll talk about flow cytometry, a brief introduction, methodology, pros and cons, and what is new in flow cytometry if time permits us. So I'll start with the first case, and all I want you to do is only register it in your minds. We'll not look at the answers at the beginning. We'll look at the answers at the end of my talk. So this is a 54-year-old female who is a resident of Nagaland, known case of hypertension with chronic kidney disease, stable on thrice a week hemodialysis since January 22, blood group was O. Of course, she has had whole blood transfusions in the past and not on any immunosuppressants, okay? So the first donor for her this is how the first donor was. You had a 29-year-old related donor who was again an O positive group. No any previous history in this donor. Typing had not been done and SAB was not available. But look at CDC, both T and B cell was positive and flow was also positive. Obviously in such a circumstance, a CDC and a flow positive, we do not really go ahead and we ask for an alternate donor. So this was the alternate donor. Now here you have an A positive getting transplanted, so that's another part of it. But again, look at it. You have typing not done, SAB not done, but CDC on T cell was negative, B cell still positive. However, flow continued to be positive. So at this point of time, the team nephrology decided that we'll go ahead and we'll look for a swap transplant. That's the other thing which people keep doing nowadays. And this is what the swap was. So we had a 57-year-old unrelated male donor. Group was compatible, no typing, no SAB, but CDC clean negative. However, flow still continues to be positive and significant, median channel shifts. Now, at this point of time, we were asked, what should we do? We said, look for again an alternate donor. That's what we keep asking. Now, they got a fourth donor. And in this donor, you had, again, no typing done, no SAB done, but CDC T cell positive, B cell negative, flow cross-match still continuing to be positive. And at this point, when again it came in, so obviously what happens, our nephrology teams generally get shaken up, especially with the kind of trust they would want to place on us, because every time they get a donor, we say positive, again positive, and every positive thing becomes negative, because then your lab is not doing your job well, okay. So this time they decided we'll go for a third-party lab, and look, the same donor when it went out, beautiful. CDC negative, T and B cell cross-match, and flow again negative, T and B cell cross-match. So the question was, do you think we should go ahead with the transplant? The question was being asked to me, and the test was not from my lab. So obviously, what do I say? I said, no, let's try and do this test in my lab again. So when we did the test in my lab again, see what happens. We again get a T and a B cell cross-match positive on CDC. We get a flow cross-match positive again. And what am I left with? Left with nothing but to ask these guys to go ahead and do a PRA SAB. Let's see what that shows, okay? So we'll not really get into the thing of this case, what really happened later on. We'll just park it in our minds and we'll come back to this case after my talk, okay? And we'll also talk about another case, which is this case now, a 30-year-old multiparous lady, end-stage renal disease, no prior transfusions, mother a prospective donor, and this I'm giving you a perspective from today's day and age, where you do a so-called high-resolution typing. You look at the, match, a proper match on the high-resolution typing. So it was a haplo match. And if you look at the CDC, it was negative, but you look at the T and the B cell cross-match, clearly positive. Median channel shift of more than 110 and 254. And of course, single antigen bead in this case, which was done class 1 and 2, was positive as well. But when you went into the nitty-gritties, you found that there were no specific donor-specific antibodies. Now, this is the second case, and in this the question is, should we be happy and just go ahead with the transplant? And how does one explain a flow cross-match positive with a SAB negative? So these two questions, just keep them loud and clear in your minds, and we'll come back to these questions at the end of my talk. So donor-specific antibody, this is what we'll talk about. And of course, we've already seen that there is enough pre-existing data available where pre-existing donor-specific antibodies actually have deleterious effects on the graft, and it's a very well-known thing. In the last talk, in fact, towards the end, it was questioned, have we really improved by doing so many gizmos and kind of assessments and tests that we're doing? Has it improved anything? The answer to that is loud and clear, yes. We have improved, because if you see these red lines which are going downwards, not only in kidney transplant, but also in hematopoietic stem cell transplants, the haplotransplants, there has been a significant improvement in the survival statistics now. And of course, the post-transplant state, we are also interested in knowing whether we have the presence of an antibody, a de novo antibody, beforehand. Again, this is something which we would want to figure out and do it quickly. Now, there are various assays and tests which my colleagues have already mentioned, which includes a CDC, flow cross-match, solid phase assays. And we know that the idea is just to make sure that we're cross-matching the two and getting the best results out. And with so many assays available, what is the best assay? We saw the CDC, we're not wanting to give up. We don't want to use the Luminix possibly because we are worried about the money that's involved. So where does that leave flow cytometry in between? Does this mean that I'll do flow and life is like glorified and it becomes the best for us to handle? The answer comes in this article, which says nothing is perfect in the art of defining HLA-specific antibodies. The moment I show you this article, what does it tell you? It tells you only one thing. If you have the possibility of doing all your assays, please do all your assays. And the reason for that is pretty simple. You have a significant variability in the sensitivity of the assays. We know CDC on one side is not able to give you a great sensitivity, but on the other hand, Luminix is highly sensitive and actually going to be making sure that you may not even do transplant in any case. So now let's change gears a little. Since my talk is clearly on flow cytometry, let's understand what flow cytometry is all about. So flow cytometry, the word flow cytometry is made up of three words: cyto meaning the cells, metry meaning measurement, and flow meaning the cells have to flow in a single stream or a flow line. And therefore, when you make this happen, the cells are interrogated by a laser, and then you get your information from there. Okay, so what happens? You have a set of fluidics, optics, and electronics, which is more of a theoretical part. I'll not get into details, but just to give you a pictorial representation, you have cells falling in a single straight line, as you can see in the laser pointer here. The laser falls on them, gives you what is called the forward scatter, that gives you the size of the cell, and on the right-hand side, you have this color thing which is coming on the right, 90 degrees angle. It gives you the complexity of the cell. And the cells are expected to flow in a single straight line by hydrodynamic focusing. We'll not get into details, but just remember, when the laser light falls on the cell, it'll obstruct the fall of the light, and because it obstructs, it causes a shadow, and that gives you relative size of the cell. And whatever falls on the right angle gives you the complexity of the cell. You're able to identify your cell spaces, the laser falling on the cells. How do we use it in HLA? In HLA, very clearly, you have the cell. On top of the cell membrane, you have these antigens. You want to identify these antigens by use of an antibody. These yellow Y-shaped structures are antibodies. And all these Y-shaped structures have a star on them, which is differently colored. That star is nothing but a fluorochrome, because laser will not light the antibody, it will light the fluorochrome. So when we put the laser light onto these fluorochromes, they get excited. Excitement makes them go to the higher orbit, but it doesn't remain for long. Nobody can remain in excited state for all the time. The excitement goes down, and a new wavelength comes out, and that's picked up by your photomultiplier tubes. And once that comes out, this signal, analog signal, is converted into a digital signal, and you're able to identify how much of a shift was there in the fluorescence. And we'll come to that a little later. So this is the cell. Any cell you can interrogate anywhere, whether on the surface, in the cytoplasm, nucleus, granules, etc. But when it comes to HLA flow cytometry, you're only targeting the membrane antigens, the antigens on the membrane of the cell. Okay, and what do? You basically get your dot plots, you create gates on them on a software platform, and then finally ascertain the presence of the fluorescence on them. So this is how it goes about. You are putting up a gate around the mononuclear cells over here, and then on T or B cells, and subsequently you are able to identify T cells and B cells very clearly, and on them you are eventually identifying presence of an antibody by way of median channel shift. So it's nothing but an antigen-antibody reaction which you're trying to figure out. And the purpose of this cross-match is pretty simple. You're only trying to identify the presence of a donor-specific antibody in an individual. The assay goal is pretty simple. You want to identify the antibody, you want to quantify it or make sure that you're getting an adequate possibility of looking at titer, and if possible, you can also look at the antigen specificity. All three things are something which you would want to do. Now, Dr. Antonio has done a fantastic job of showing you how CDC gives you a lot of information, and I'm not here to say that CDC should rest in peace. It has its own role and status in the system. But why should we talk about flow cytometry? Is because you have low sensitivity of CDC, it's a relatively more sensitive and a quantifiable methodology, and it does not have inter-observer subjectivity. Somebody was asking from the audience earlier that if I see, a cadaveric transplant, I get more cell death. Well, that's a fact of the matter of life. You do get more cell death in that, and looking at the plain tube again becomes equally important. And of course, when we get to the procedure, we do it almost on the similar lines like a CDC. I'll not get into details for the want of time. But remember, we need to get the donor sample, which should have the mononuclear cells, the lymphocytes, serum from the patient, and of course, the negative and the positive control. And all this needs to be put into into the tubes, which needs to be put into the flow cytometer. The only part of it which I would want to emphasize on is to identify the antibody. We need to make sure that we're using only the FAB fragment of the antibody for our flow cytometry. The reason for that is if you have the FC portion of the antibody, the bit of the Y, which is there, it has a probability of getting fixed to monocytes and other mononuclear cells non-specifically, and that can give you a false positive reaction. All right, the other part of it is pronase. Somebody was talking about in the morning, do you use pronase on CDC? A usual concept is no, but sometimes people do use pronase. We use pronase upfront in our flow cytometry, and this also helps us in cleaving the CD20 molecule so that you have a rituximab effect, you can take care of it. But remember, when you do it for the cleavage of CD20, the concentration of pronase to be used is higher, and therefore standardizing it can become a potential challenge. Anyway, so what do you need? You need serum from the recipient, you need cells from the donor, but you also take cells from the recipient to put up an autocross match. And that's why you take these samples, you call it down, in a usual circumstance, take the lymphocytes, put them to reaction with the recipient's serum, and then finally when you've done this, you're able to see whether there's a median channel shift, significant shift or not. Now, life was bliss till I was using call, and just about last year, early last year, we realized that many a times when you use call, you're not getting rid of all the other cells other than the lymphocytes, because lymphocytes is what you're concentrating on. So that's when we decided that we'll move on to something called immunomagnetic separation, which Antonio has anyways started in his center already. The reason is that when you use this methodology of positive or negative selection of lymphocytes, you are obviating the possibility of a false negative flow cross-match. Because again, the antigens are not only present on lymphocytes, they're also present on other cells. And if you have a weak antibody present in the serum of a patient, it will not bind only to lymphocytes. It will bind to the other cells which you are not interrogating. So therefore, this methodology of using what you call positive or negative selection is what people use. We use a positive selection because it's cheaper that way. And this is the magnet which he already showed you. You can use automated mechanism also. And then finally, when you're getting your cells, they're pure cells, pure lymphocytes which you're working on. I'll not get into details of this, but just look at these two graphs, and visually they're pretty appealing. If you see the top graph over here, you can see these green cells over here, green and red mix and blue mix, but you also see a lot of black cells on the top, whereas when you see the low one, you hardly see any of those coming in the assay. And the moment you see them, you realize that your lymphocytes are pure compared to the here, but here you do not have only pure lymphocytes. So therefore, positive reaction becomes much more pronounced in that. And the same is also depicted graphically over here. I'll not get into details. Now, this becomes extremely important when you're doing cadaveric transplant, because in cadaveric transplant, by virtue of the fact that you're taking sample from a dead man, you have a lot of cell death already, which is coming in the background, and that does not leave you with a lot of good cells to work on as far as flow cross-match is concerned. Additionally, there's also a lot of non-specific binding of the antibody that you're trying to look for. So therefore, going across and doing a very specific lymphocyte separation by using positive selection, you're getting a much better reaction. Quickly, let me try and show you how it works. So if you see, we're using CD3, CD3 is an antibody which identifies my T cells. The white one, which is here, is the antibody which was present in the serum. I use an antibody which is against IgG with a fluorochrome tag to 𝒏𝒂𝒏𝒂𝒏, and this gives me a positive reaction and identifies T cell reaction. Similarly for B cell reaction as well. And of course, we can also do IgM part of it if you want to identify IgM separately on flow cytometry. We do it in our center, most of the people don't. And this is the usual flow cross-match protocol which most of the people use. Again, for the want of time, I'll not get into details. We use about a 10-tube protocol. Needless to say, calibration becomes extremely important for people before you get into a flow cytometric assay. And you also need to remember there's something called a median channel shift with a negative control that you have to be taking care of. Now, what do we follow in general? This is the median channel shift which people talk about. On a flow cytometry, you have two types of scales. One is called 256 scale, the other one is 1024 scale. Depending upon what scales you're using, your cutoffs will be different. And that's why a lot of people prefer to use a ratio instead of using the cutoffs, which perhaps has its own advantage. And again, the interpretation of flow cross-match will differ depending upon how you are using it in your own settings. But for all practical purposes, if you have a positive reactions in either of the two, be very careful and mindful of the transplant you're going to take ahead. Now, this is a classical picture of a flow cross-match in today's day and age. I'll show you what we were doing conventionally also. You use a first dot plot, which is called a time dot plot, to identify whether there was any break in the process of the sample when it was being put across. You take singlets because you don't want any doublets to come in and interfere in your assay. Then you take only the non-debris events, these are the debris events which you don't want to interfere. And then once you have assessed that, you take the mononuclear cells, and on them you identify the T and the B cells, and then you identify whether there was any reaction or not any reaction. So usually a negative one will always show you the spike in the first log of the graph itself. This is how it was when we were using phycol. Just look at this. Not only did we get mononuclear cells, which you're seeing over here, we also got these debris. And of course, these two cells which were distinctly there. And this was a negative one. Now let's look at the positive case. Just look at what has happened. The peak used to be here. It's moved significantly rightwards. And this significant rightward shift is what is called the median channel shift. The reason why we use median is because we don't have a very classical bell-shaped curve here. We have a very zigzaggish curve. And therefore, median is what is preferred and not the mean. And this is how IgM part of it goes. I'll not get into details. Again, IgM part of it is seen the way it is seen in this particular picture over here. Now, when you interpret flow cross-match, it becomes important for you to know how you'll interpret it. Just look at this particular picture in front of you. You know, on the red part of it, the red peak is actually your classical negative sera. But you can have samples which can have all different variabilities with respect to positivity, with this green picture being very strongly positive. So interpreting these small, short negatives again becomes crucial. And how do you decide this is by setting your cutoffs. I'll not get into details of setting your cutoff, but remember, having a very high positive reaction, diluting it like a serial dilution and getting across to your assay becomes equally important. And this is how eventually this will pick up. So you on a T cell, you have a classical positive reaction, a classical negative reaction, and this green one gives you a positive case. Again, on a IgG, on a different case, you can also get this kind of a reaction. So variability in expressions is also something you need to keep in mind. So all this we've been doing for lots and lots of years. This is the data from our current place of work. We have almost about 1,500 odd flow cross-match cases. You can see the positivity, the red bars down here. And what you would notice is 2021. This obviously was the COVID year. But interestingly, in this COVID year, we had a much higher positive reaction. God only knows why. Maybe only 9 cases, and probably to interpret from 9 cases may not be right. Or as everybody else says, COVID was a big, interesting mimicker, and it would give you lots of reactions likewise. This is what we got in positive cases. We got almost 85% IgGs, and the numbers were not too many. Out of these 1,400, 209 cases, and 15% were IgMs, which eventually got resolved, and there was no class switch in this case. Okay, so that gets me back to my cases. I am possibly just in time. So this is the same case. We go back and revisit it again. Again, the same case where we had too many donors and, swab donors and stuff like that. And then we had a lab from outside giving you a clean negative, clean chit, asking whether we go ahead with the transplant or not. And then we giving you again a question whether this is still positive. Let's do a PAB, SAB, and a PRA. Now, in this particular case, if you see, both PRA as well as 𝒌𝒏 were positive. And in this case, even the 𝒌𝒏 was very clearly and distinctly showing positive reactions. The question was whether there was relevant antibody or not. In this particular case, it was significantly relevant. It's a different story. We didn't go ahead with the transplant, and in any case, the patient succumbed later on, just about 2 months later. But nevertheless, the idea in this case to tell you was that please don't ignore what is coming from different tests. All tests will give you different reactions, and there will be variability in reactions depending upon who's performing the test also. The other one, which was where high-resolution typing had been done, you had a 6x12 antigenic match, and then, of course, you had no donor-specific antibodies. But if you went back and tried to look for it, you realized that there was a particular allele which was not represented on the assay. And this was something which has already been reported by lots of our colleagues. The first one here is from Vellore, the other one is from Mumbai, and of course, some of us have similar experiences in the past. And if you look at this one, which is here, which is from Mumbai, where they've shown nearly 13% not being represented in one panel and 25% in the other. And of course, internationally also, by the way, in the Asian part of the world, you still have significant percentage which is not represented. In fact, the one from Vellore showed almost about 50%, if I'm not mistaken. So challenges of unrepresented alleles in the 𝒌𝒏 also become equally important, and thus becomes crucial for us to understand. So a combination of flow and cross-match is what I would recommend for all of you to review, think, and not only depend on a CDC or only on a flow or only on a 𝒌𝒏 and stuff like that. Develop an algorithm in your system. We have an article. For the want of time, I will not be able to get into those slides. But if we follow an algorithm of following a system of at least one cell-based cross-match and another bead-based cross-match, and then interpret and then go ahead, it might give us a better advantage of identifying the antibody, pre-existing antibody in an individual. I think I'll stop with this because of the want of time. Should there be any questions, we'll be happy to answer.
Thank you very much, sir. It was very enlightening, and it was very brief and short. You could cover almost all the topics. So with the more information comes the more confusion, so which you highly rightly pointed out. So just for an understanding, so when the flow cross-match shows some positivity and your 𝒌𝒏 is negative, so will you work up further in these cases?
So whenever we will have a 𝒌𝒏 negative, the first thing that we would try to do is this. We'll try to find out, do we have the representation of the alleles in the 𝒌𝒏 or not?
Yes, sir.
If there is adequate representation of the alleles in the 𝒌𝒏 and we have a negative cross-match, then we would not worry really much about it. But if we do not have representation, then we would take cognizance of the flow cross-match and we would rethink about strategy of how we'll go do desensitization and only then do the transplant of that patient.
So just one more question. Like when we are doing dilutions in flow, so how does this mean channel shift, like median channel shift, actually, will it be proportionally adjusting itself, or it is like we have to do some correction factors in that, sir, when we are diluting?
So it's a very good question, and I would have expected another question to come on, that is MFI, and somebody might be talking about it. So they say MFI is mostly fluid idea. That's what somebody has said it in one of his articles. And when we talk about median channel shifts, you say mostly it doesn't remember where it has to shift. Now, when we do MCS and we do flow-based cross-matching, when we do dilutions, remember there will not be a proportional linear kind of a relationship between the two. So when we do flow cross-match, we have to give cognizance to the fact that we must put up dilutions upfront. Some of the centers like us, or maybe even Vellore, we put up dilutions upfront, which gives us an indication whether we have a prozone phenomena affecting the assay or not. And that's a very, very important thing to do. Because if you have a prozone affecting it, your neat assay may not give you the reaction at all, but a subsequent dilution will pick it up. But remember, it will not be linear.
Linear, okay. Thank you very much, sir.
Question is open to the floor, please.
Hi, sir. That was a beautiful presentation. Coming to the 𝒌𝒏 antigen presentation in the all of India, is it homogeneous? Does it vary from state to state? Has any studies being done on that? Or are you just depending upon imported antigens?
Okay. So there are some studies currently underway, if I may say so. I think Vellore is also working on that. There is one center in Delhi working on it as well. The issue here is with whatever little information that we have in the last almost a decade or so on single antigen bead assay kits that are available, there is underrepresentation of beads of our population, which was emphasized even in the first talk today. Thus, calling SAB as a gold standard would be preposterous, especially in the settings that we are placed. The second part of the thing is that should we not then try and start developing a kit of our own, where we should have significant antigens which are present in our system here in our part of the world? The answer to that is that yes, there are some people who are trying to develop that. There is in fact one of the companies which has been given a DBT grant for developing it, and they are currently kind of working on it. I happen to be associated with it a little, but I don't think it's going to be coming up in this year or next year. It's still going to take at least a year or two to really come into the market for commercial use. But having said that, our population is just a bit too diverse, and getting hold of all the antigens again is practically impossible. The alternates which are available to us as on today is the epitope matching, which unfortunately has still not found its kind of roots, and I'm sure you are talking about it, Feroz. So Feroz will give you a little more detail about it, which will make you understand why we should start looking at epitopal matching rather than just asking for a SAB and just doing a high resolution typing.
Any more questions? Yeah, please, Praveen.
Basically, we started flow cytometry lab recently, and we had a lot of difficulty in sort of standardizing it. Is there a method, because there are so many machines and so many people starting to do flow in different parts of the country, is there a method we can sort of standardize the way, because you showed the first case where the outside lab was showing a negative and yours was positive. And that is really unfortunate because it's basically a wrong report. So is there a way of standardizing this in our country, sir?
Okay, so there have been efforts in this direction for the last more than a decade now. There are two booklets which are in fact available online. One is from Medanta, where we had done the first workshop where we had set it up as an assay and defined as to how to make sure that everybody should follow the same methodology. Remember, when we talk about flow-based testing, it is a lab-developed test. There is no kit available in the market. So you don't have a kit-based assay, which is designed, defined, validated. So when a lab-developed test is designed in any hospital, in any lab for that matter, you will have to make it undergo through the entire process of validation. And generally speaking, if we follow what our predecessors have done, invariably you will end up doing the right thing, because that has already been kind of published and it's already available in literature. So similarly, again, there is a standard, again, with respect to flow cytometry, setting up a flow cross-match that's available from 𝒐𝒊𝒏𝒈. It's in the 𝒐𝒊𝒏𝒈 manual. If we follow that religiously, exactly what they've asked us to do, I am reasonably sure you'll get consistent results. So as long as we are following some described literature, we should be able to get what we're expecting. The only thing is that people do not ask for dilution, most of them. What we recommend today is that at least you should have 2 dilutions, if not more.
So as Dr. 𝒕𝒊𝒆𝒅 was saying, we have come from a stage when we have started with CDC, then we have come to CDC flow, single antigen bead. But when you're working in a resource limited settings, when you have limitation about, I understand the transplant is it's a costly affair, 50,000 this side, that side doesn't really matter. But when you're really working in a bedside, it does matter. So do you, as I understand is that you told one cell-based assay and one bead-based assay. So, but most of our centers, what we do is we do a CDC, then we do a flow. And if we find that CDC flow is negative, then we feel safe. Do you think that approach is better? Or should we do a CDC and then go for an HLA screen using a bead and then go about it? So I understand there is a flow chart, but as per your experience, what do you think is a better option?
You put me in a very difficult state. If I say anything, it should not be taken as gospel truth. Huh? The only thing what I can tell you is, as far as CDC and flow cross-match is concerned, if you would have asked me this question maybe about 8 years ago, I would have said, life is good. CDC flow negative, go ahead and do a transplant. But I have burnt fingers after that, so I know that CDC and flow alone are not sufficient for you to catch hold of a lot of antibodies. And sometimes, because again, if you remember the first talk, it was very clearly mentioned that at a certain MFI level, flow will be positive only after a certain MFI level. And similarly, CDC also will be positive after a certain MFI level. That's why if you go to consensus recommendations globally, one thing is for sure, at least one bead-based assay is what they're expecting you to follow, and one cell-based assay is what they're expecting you to follow. And even when you go to the recommendation that what we have given from our group in Delhi, where Mohit and Aseem and I were together, we realized doing one cell-based assay which is preferably a flow, and then you have another bead-based assay, which is an antibody, HLA antibody screening assay or a PRA assay, whatever you want to call it. If you are getting negativity on both, you're reasonably safe to almost about 85 to 90% cases, and you will go scot-free as far as transplant is concerned. That's why we keep recommending, and the cost of both of these together will not cross you more than maybe 12,000 rupees. And therefore, this whole activity of 12,000 bucks will actually help you in making sure your transplant is successful. I think that's one message which should go very clearly from today's lecture.
Since we have run out of time, I think we need to close this session. Thank you very much, Dr. Handoo. That was a very nice
Thank you,
talk.
everyone.
Yeah.
Thank you, sir, for that insight.